Biology operates at blinding speeds. Inside a single cell, millions of protein structures fold and interact in fractions of a second. But as our ability to capture this staggering amount of data has scaled up, human cognition simply cannot process it all at the same pace. Managing fragmented digital tools has become a primary bottleneck, consuming the time researchers once spent on actual experimentation. Stitching a fragile pipeline by hand, pulling from PubMed, coding in Jupyter or R, and bouncing to a remote cluster via SSH takes weeks. Forcing these databases to talk guarantees one critical thing: absolute granular control over every single step. Because of this friction, highly trained researchers spend the bulk of their day acting as pipeline constructors and IT managers. The actual work of testing scientific hypotheses gets pushed to the margins. Anthropic recently introduced an alternative approach with Claude Science. They call it an AI Workbench. It replaces that fragmented manual toolchain with a single natural language interface. Instead of you manually converting files, the platform natively integrates the tools and packages you already use. It manages the file formats, sets up the bespoke data pipelines, and queries the databases automatically. For a bench scientist, this immediate automation raises a question of rigor. Handing pipeline control to an AI agent can feel like a threat to the strict reproducibility required for peer-reviewed science. That is the core trade-off you have to weigh. You are trading direct, granular control over your code for a massive increase in speed. The challenge is ensuring an agent-driven workflow still produces the auditable, publication-ready results required for peer review. Let's track these two approaches on a comparison matrix, starting with the first phase of the workflow: knowledge synthesis and database querying. The manual method requires you to individually cross-reference sources like Uniprot or Kemble. Each has its own specific schema and query language. You get exact precision, but a human can only read and process a tiny fraction of the available literature at a time. Claude Science handles this differently. It uses over 60 pre-configured connectors and specialist agents to query thousands of papers and databases simultaneously. The scope and speed are massive, but it forces you to implement strict verification methods to catch dropped citations or errors. A neuroscientist at the Allen Institute built a workflow specifically to manage this verification problem while writing multi-study reviews spanning over 100 pages. His team uses an actor-critic multi-agent architecture. One specialized agent reads the papers and drafts the narrative section by section. A completely separate reviewer agent is deployed purely to evaluate that content, strictly enforcing accuracy and citation fidelity. Using an agentic workbench changes how you interact with the literature. You spend less time reading every primary source manually, and much more time acting as a strict auditor of the synthesized outputs. The second phase of the workflow is compute and pipeline execution. Normally, running a heavy genomics pipeline requires you to write custom bash scripts, manually authenticate your remote SSH logins, and then wait your turn in the queue on your lab's high-performance computing cluster. This setup guarantees that your data is secure and gives you cycle-by-cycle control over the hardware, but it also creates a steep technical barrier for bench scientists who aren't trained in system administration. Claude Science takes over these logistics. It drafts a compute plan, asks for your explicit permission, and then routes the job to your local lab infrastructure or scales it on demand via a cloud service like Modal. Because the agents work inside a running session that holds context in memory, you only have to load massive datasets once. You avoid the wait times of constantly pushing and pulling data across networks. Adopting this agentic compute model strips away the setup friction that slows down daily research. But in exchange, you have to entirely relinquish direct oversight of the underlying pipeline execution mechanics. The final workflow phase addresses the most critical standard: artifact generation and reproducibility. In a standard workflow, you manually code your figures in R or Python. If you adjust a single statistical parameter early in the process, you have to manually recalculate and re-export every single downstream chart. Claude Science natively renders complex scientific artifacts, like interactive genome browser tracks or 3D protein structures, directly alongside your manuscript text. To satisfy the strict requirements of peer review, the platform attaches a persistent history to every single output. When it generates a figure, you get the exact code, the environment details, and the full message history that produced it. If a visual needs adjusting, say you want to strip out the grid lines or change an axis to a log scale, you request the edit in plain language, and the agent rewrites its own code. To hit the quality threshold for a publication-ready result, your core proficiency has to shift. You spend less time memorizing raw plotting syntax and more time mastering how to iteratively refine your prompts. An agentic workbench functions as an abstraction layer over research logistics, requiring the same level of scientific rigor as any manual pipeline. This chart plots time allocation. For inventing novel computational methodologies or bespoke schemas, stick to the manual toolchain for absolute control. But for compiling massive multi-study reviews or running established CRISPR pipelines, the agentic approach offers a massive advantage in speed and scale. Deciding which tool to use comes down to your lab's operational priority. Are you trying to invent the pipeline itself, or do you want to rapidly execute established methods to find an answer? Giving up granular control of your daily digital plumbing buys back the time you need for high-level synthesis. By lowering the technical barrier to data wrangling and complex coding, sheer technical prowess will no longer be the primary factor separating one lab from another. As technical barriers to data wrangling drop, a lab's output depends more on the strength of the original hypothesis and the researcher's ability to orchestrate these specialized agents.